Alrighty. Well then, okay. So I'm Jen. I guess my talk will be a little bit different now that I know that there's just us. So interrupt me, and and we'll talk about whatever seems interesting to you. But the general topic is drug target identification and translational research that we're doing here at Penn using RNA sequencing in single cells specifically. But then I'll talk about about RNA sequencing, not just in single cells, in tissues and other things. So transcriptomics is the study of expression levels of RNAs produced by the genome in a sample at a given time. So DNA, a segment of DNA through transcription will lead to a bunch of copies of an RNA, and then that single RNA through translation will lead to many copies of the corresponding protein. And transcriptomics is just focused on this one part, the RNA, where you're taking a snapshot, and this is sort of the output of it. Each column represents a different sample, and each row is a gene, or an RNA, and there's 30,000 RNAs, so this is just a couple dozen, but you're showing abundance of each gene. So blue is low abundance and red is high abundance. And when you look at each column over all 30,000 and you look at the abundances of all of them, that's called the expression profile. And so what can we gain from studying expression profiles? Well, we can identify previously unknown genes. So if you have a heart sample, you would expect to see genes related to receiving input from the body and deciding if it needs to be faster or slower, depending upon your activity level. And since the heart's a muscle, you're going to need a lot of energy, and energy comes from mitochondria, so you're going to expect to have a lot of mitochondrial genes. And so that's all good, although you wouldn't know how abundant those ones are. So we can find all that out. But then also there's just lots of genes responsible for making the cell function and receiving input from other parts of the body that we don't even really fully understand yet. And all those genes are there, and we can find out which ones they are and how much of each one there is. So this is a figure from a paper that we did semi-recently, and these are samples from two different subregions of the same region of the brain called the dorsal raphe. And these little spikes are action potentials, and they're sort of a measure of how active that region is. And so in this part, the VM, not too much activity. In the LW, lateral wing, they're just spiking all over the place. So they're just a lot more active, but nobody really knew why. And so looking at RNA sequencing, we could look at some VM cells and some LW cells, and you can see that with these genes, they are just so different. And then if you focus on the exact genes, you can start to piece together pathways and see, oh, in the LW, this pathway is just extremely strong, and in the VM, these pathways are extremely strong. And so if you're trying to target them with drugs, for example, you're going to want to use things that target these genes for the LW and these ones for the VM, and I'll talk more about targeting them later. You can also find differences between diseased and normal cells, which I'll talk about a lot later. This is where you come into translational research and personalized medicine. If you can look at the diseased cells and the normal cells from a single patient, you can figure out for that patient why their diseased cells are going so wrong, and figure out treatments specifically for them. And then, also a lot of researchers study development. So you can take samples from an adult mouse, a particular brain region of an adult mouse and of a cute little baby mouse, and get all the ages in between, and you can see how genes change over time. And so that also can relate back to medicine. You know, for a child, the disease is different. You're gonna wanna treat them with different medications than for an adult. So, you know, it's a couple steps away, but this is a really important step.
And this kind of means to have, within 60 minutes, do something or something with the medication between males and females.
Absolutely.
It's huge.
Totally. Absolutely. And I mean, researchers do a terrible job. Terrible job. You know, it's easier because to get rid of variability, you choose one gender and you stick with one because it takes less time to do both than to, you know, do a thorough study of each individual one. So you choose the male because the male is, you know, doesn't have cycles throughout, and so you choose that one, and then the female gets, you know, we get a drug that was designed for, you know, male disease. So yeah, absolutely. And those are studies that, you know, through cycles, through ages, different genders, all this stuff can be evaluated using RNA expression profiling. And the great thing about it is it's really high throughput. So that's, studying protein this way is harder because there just aren't currently as many assays. We've been working really hard to make a huge selection of assays to study RNA level. And hopefully we'll get to that point with protein, because ultimately it's the protein that's functioning in your body. You know, the RNA is leading to the protein, so it's great. But you know, ultimately, hopefully, we can do these kinds of studies on protein as well.
Can I back up a minute and just speak to the disease and normal cell? Mm-hmm. And it kind of tags on this and what I've been hearing since I've been here. The variations of what we have to be able to make a template to say, well, this is a disease cell compared to something that is necessary at this point in making that makes this particular individual, which goes into a broader thing, if that makes sense. Because I don't know, when you say disease, is disease normal? I mean, disease is something that is universal across, or is that just based off of what has been defined so far? It isn't normal to the regular things that have been identified.
So there is, so if you have a person who has a disease, there's a chance that all of the cells in their body will be affected by that disease. So even if you take undiseased area from that patient and compare it to their diseased area, it'll be different than, hopefully, my undiseased area. So are you asking about deciding on what to compare?
Right.
Yeah, so I mean, the great thing about this is you can kind of compare almost anything, and with these, since it's so high throughput, there's banks of tissue, which I'll talk about later, where you can survey many patients, and so you can kind of find the variability that is normal, and then find the, when I say disease, I'm talking about a particular disease. I'm not like lumping like, you know, diabetes in with cancer. But like when you look at diabetes, let's say, because cancer even is, you know, goodness gracious, you know. So diabetes, no, let's pick breast cancer. You know, so you can find breast cancer patients, and you'll find the cloud that is the cancer cells and the cloud of normal cells within those patients. And that kind of, I'll talk about it a little bit later, but when you study single cell variability, you can find that cloud of normal, because this is not normal. This is normal. Like, you know, everybody's different, and all your cells are different within your body. Even, you know, a liver cell is not a liver cell is not a liver cell. There, it's a cloud of grouped into liver cells. So yeah, then another great thing, which I'll talk about a little bit later, kind of in depth, is finding the effects of drug treatment. So you can find the RNA expression profiles of diseased tissue, and then treat, and then get another expression profile. See if it's close enough back to normal for your tastes, and if it's not, then give another treatment, and then test again, and see how, combinatorial treatments are the way to go. Like not one treatment works for everybody, and it's probably a combination of things that are really gonna solve the disease. And it can be used for so many other things, but those are the things I listed. So to do transcriptomics research, these are the general steps. First, you need to harvest your sample. The way we do it, and this is what I was talking about, this is what the kids did when they came to our classroom. These are primary neuronal culture, and so you can see these little neurons, and you come in with a little pipette, so it's a capillary tube that we've like pulled to an extremely fine point, and you come in and you literally suck them up into the little pipette, and then you can see that only that cell was collected. These other guys are undisturbed, and these kids come in and they do it, and it's awesome. Or younger. Oh, high school kids easily, and younger too. Yep. Yeah, it's pretty.
And this isn't your nano division, that's something different.
I'm sorry?
This isn't you. I know the kids come in and do some nano related things. That's not. You're just saying, okay.
I don't know what the nano kids do. Yeah, this is my boss does a lot of outreach, and he and they come in and they're so excited, and they do, and you can take these pictures, and they do a great job, and so it's pretty neat. And then I do it too, and this is how we actually run our experiments for our single cell work. You can also use LCM, which is laser capture microdissection, which is another technique. You put a plastic film kind of over, and then you apply a laser and sort of circle the cell or tissue that you want, and it gets stuck to the capture device, pulled away, and then deposited elsewhere. And so you can see you've now captured this and left all of this, so like imagine that this is the tumor, and this is the surrounding tissue. You can study the tumor, you can study the surrounding tissue, and
then even like the borders and stuff.
Oh yeah, you can. And you can chop this in half. I don't know too much about this sample type, but imagine if this little thing is very important, you could get just that too.
Wow. And that's LCM?
Yep, LCM, laser capture microdissection.
Who makes that?
I don't know. We don't use it because we have one in our lab, but I don't remember the name of the company. We do a lot of this and more advanced techniques, but these are great. So this is in culture. This is in fixed tissue. So ultimately,
you said single cell or even.
And then ultimately, what our lab likes to do is collect samples from fresh live tissue. So you can bubble the tissue with oxygen and give it nice nutrients and keep it very happy, and then you can collect the cell in its natural microenvironment. So it still has all those normal connections with everything else. Like these guys, they're good, and they have connections with these cells, which keeps them generally happy, but this isn't the cell that was next to it in that mouse's brain. You take the tissue out, you break it into the individual cells, and you plate it out. And it's good. This is great. You can get a ton of information from this, but ultimately the best is to get it with all of its normal, natural connections so that it's unaltered. It's one step removed. But at the same time, you can do an awful lot of stuff with culture that you can't do with, a chunk of tissue. And also, we can have a lot of this, whereas, if I'm working with patient tissue, it's precious. It's extremely precious. So we don't have as much, and we can't do as many experiments. So it's depending upon what you're trying to do. There's different methods. So since we're collecting such small amounts of material, you have to amplify the RNA, which I'll get a little bit nerdy and talk about in the next slide. And then you have to prepare the RNA to go on the sequencer, you sequence it, and then you get just an insane amount of data. And this is something that I was thinking about for projects for kids. I mean, if you use Excel, you can do some of this very basic analysis and have a real world meaning behind it, you know, as opposed to just adding, you know, different things. So we could talk about that later if you'd like to talk about it. And then, so we have the expression profile, and then, so since it's RNA, we like to follow it up with some more experiments to show that what we're seeing at the RNA level also occurs at the protein level, and then any number of experiments after that. So this is the amplification protocol that we use. So we'll get real biology-ish. We have our mRNA, and mRNA has this poly-A tail on the end. That's what it always has. And so we use this primer that has T's that are complementary to all the A's, and then we've hooked this T7 promoter on, which I'll talk about why it's there. Then we fill in the ends, and we have this double-stranded thing with our T7 promoter, and then the T7 enzyme sits down here, and it reads along, and it makes a copy. And then another enzyme comes along, and it goes, reads along, and it makes another copy, and da-da-da-da-da. So, and the great thing about this is, since it's always going back to this original template, the ratio of all the RNAs is maintained. So if you have one RNA of RNA A and 2 of RNA B, you're going to, you know, if you do a bit of amplification, you'll have 2 of this and 4 of this, or so on and so forth, depending upon how long you let this reaction go on and on. And so all of the ratios are maintained, but you just have enough of it to be able to do some meaningful analysis on. And then so RNA, there's some little RNAs and some big RNAs, and so after the amplification, we use a bioanalyzer, which basically we use to find out the amount of RNA that we have and how long they are. So this is a marker, but so we have a little bit of small stuff, we have a whole bunch of middle-sized stuff, and we have, you know, enough big stuff. And so this is sort of the distribution that you would expect of different sizes of RNAs in a cell.
So this is, like, this is happening in vitro, but in vivo, this would be simulating the transcription phase where the RNA is reading, and then the copies and copies and copies is the translational. So it's IVT, in vitro transcription. So because we've turned it into cDNA and we're making RNA off of it, it's transcription. Okay. But yep, yes, exactly. I mean, it's similar enzymes doing similar things, you know, reading along and matching on complementary bases. So once we have our amplified RNA, we fragment, and then we add on these little adapters, which are necessary for when we put it on the sequencer. And what happens is we have this flow cell, which is just like a solid thing, and these little adapters bind on, and an enzyme comes along and it says, oh, there's an A, let's put a complementary T, and it blinks blue. Da-da-da. Oh, there's a C, let's put on a complementary G and blink green. So it goes blue, green, orange, blue, purple, blah, 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 and so on and so forth. And through fancy computing, it says, oh, okay, that was GTCTAC, and that is a read. So we have all these reads, and then so for a mouse experiment, for example, we know all the genes in a mouse. We know the transcriptome. So it takes a single read, and it looks along the entire transcriptome, and it says, oh, it fits here. And then it takes another read, and it looks along the entire transcriptome, and it says, oh, it fits right here. And you keep going along the whole transcriptome. And so this is the bioinformatics. So this is like the mathy part of this, which
I hear a lot about this field. I mean, that's something
so there's all this big data. There's genomics, there's transcriptomics, there's just data. And there's people that need to know how to deal with it. And we have wonderful, wonderful people who can write programs, but they also need to understand the biology too. And, so they're special, wonderful people.
Yeah.
And with these huge data sets, I mean, there's a huge demand for people who can handle this.
I've never heard of that until maybe 2 years ago in this field.
I can try to scrape by and do some stuff, but what they can do is amazing. Mm-hmm. So collaboration is very important. So they're awesome. So imagine all the reads piled along the whole transcriptome, and so we're going to focus on gene 1 and gene 2, and then we have 3 samples. And in sample 3, there were some reads that fell on gene 1 and a whole ton that fell on gene 2. And you can quantify how many reads stack up and correlate it back to the abundance of that gene in that sample. And then from that, you can generate these heat maps. So for example, like sample 3, a little bit of gene 1 and a whole bunch of gene 2, that would be like this and this, a little bit of this and a whole bunch of this, and all the way down the transcriptome, all the genes, and then you can compare all your samples. And you can cluster them. So you can see these 2 samples seem more similar than these 2 samples, although they're not identical. So you can see like this guy, it's a little bit lower abundance than this one. Even though these 2 cells are the same cell type, there's variability. These are single cells. I always talk about single cells because that's what we do. But this kind of thing, well, actually, I'll talk about it in my next slide. This kind of thing just was not appreciated before single cell research. You take a hunk of tissue and you analyze it and you say, okay, yep, that gene is highly expressed in that tissue. But it's not quite that simple. So these are my 2 tissue samples of 5 cells each. And in this one, if you squish all these cells together and you analyze them all at the same time, unfortunately, you might miss these red triangle genes because with this type of research, you need to set a threshold. There's some sort of noise and you just need to set a threshold above which you believe the data and below which it's a little bit iffy. So you might miss these little guys and you might say, yep, they're all white squares. Alternatively, in this tissue, you squish it all up and you kind of get an interest, you detect both, but you're missing the whole story. I mean, this cell has no red triangle. These cells are almost exclusively red triangle, and you had no idea when you squished up just that whole piece of tissue. So by doing single cell work, I can now see that, I might say that, okay, there's more white square in this tissue. We should target this for our treatment. But I would totally be almost missing treating these cells, and maybe these are the important cells that we need to be treating. So single cell is.
It is interesting too because we certainly don't teach it that way. I teach, I was just going over sexual and asexual reproduction, mitosis. We teach their exact copies. Mm-hmm. And with the variability, although exact copies might not necessarily mean that their expression, right, they could be or couldn't be when you just showed that 2 slides ago. Mm-hmm. Again, not that the one before where cell 3 and cell 4, so they're mm-hmm. identical, yet the expression is different. So
yeah, they're identical traditionally speaking. Okay. I would never say any cells are identical.
Yeah.
Yeah,
no, but I think that that's really something that
it's not appreciated. Yeah. It's not fully appreciated, and it's, I think, critical for cancer research, which I'll talk about later.
I was thinking that, exactly, because there's a point where it's no longer, normal or functioning normally, and then something gets cut off or turned on, and where did that actually happen?
So these are just some of the different cell types that we've done single cell sequencing on. So this is our bread and butter, our primary neuronal culture. This is a project that I'm currently working on. It's brown fat. Brown fat is involved in regulating your temperature of your body. It's involved in fever. It's also hugely involved in obesity, diabetes. So it's a really important thing, and next to nothing is known about them. Over the past couple years, there's been some stuff, but we just don't know enough, and we could help so many people if we figured out what these little guys were doing. These are just pretty. We've done these dinoflagellates. This is getting to what I was saying earlier. This is we took a mouse brain and we sectioned it, and we kept that slice alive. And so we did the measurements of the activity of those cells, and then so we patched in, we connected with the cell, measured the activity, and then sucked it up. So we now know how that cell functioned when it was still alive and in contact with all of its normal contacts, and now we know the expression profile too. So it's just much more powerful. We've done taste buds, we've done fly brain. So fly brain, it's just a really good system to work in. You can do a lot of transgenic work. The whole brain is sort of mapped up. Everybody understands how everything's all connected. So you can come in and harvest. Again, you can measure the activity of the cells and then suck that cell up. And we harvested a bunch of these cells in relation to a circadian rhythm project. Flies are just pretty handy to study a lot of things in. And then, this is just related to this. It was a transgenic mouse that had these particular serotonergic cells fluorescing yellow. So you can see that nice yellow cell in my little pipette there, and I just sucked it up. So.
Do the brown fat cells have anything to do with inflammation, study of it? Because when you said fever and obesity, I was wondering if there was
I'm sure that it's all related. Fever and inflammation kind of go hand in hand.
Inflammation is the cause of so many, inflammation that you don't even know.
Mm-hmm. So you lose a lot of your fat when you get older. So maybe they are involved in. I'm totally,
I was just wondering if you knew.
I don't know particular.
I've never heard what brown fat even is. Never even heard.
Yeah, children have a lot of it, and then you have less, but it's still. It wasn't fully appreciated how. People thought that it wasn't in adults, but now they found that it is, and it's pretty important. And some anti-obesity drugs, actually, sort of the main one targets before our study, the one receptor that was known on these cells, and it was good. But my paper that we're sending out now shows that there's a lot of other receptors that we could be targeting that are actually more evenly distributed in all the cells. The target they chose, because they took brown fat and they squished it all up and they said, oh, yep, there's a lot of that. When you look at the individual cells, it's highly expressed in some cells, but not in all of them. So we found some targets that are more evenly expressed, so we can target all of those cells for maybe an even better response. So single-cell transcriptomics really lends itself to translational research and personalized medicine. So I was mentioning earlier, there's these large public banks of patient tissue. So you're not limited to, what's available right now. People who donate tissue to get involved in clinical research or donate to, their bodies to science. It's just such a spectacular resource. Most of the research has been working in, mice, and then or flies, and then mice, and you work your way up. This resource is skipping straight to patients. it just makes everything faster and less failure. It just requires an amazing amount of appreciation for what these people are willing to help us with. But so this is a biopsy, and so you can see, this is a mouse, but you can see that these are the normal cells and these are the cancerous cells, and so you can come in and you can isolate each of these different kinds and sort of see that sphere of the cancer cells, what the cancerous cells are, and the sphere of what normal is, and find what targets are in this that aren't in this, so that you can hopefully only affect this and not affect this, because cancer drugs now, you're kind of affecting everything.
Question comes on, since I guess a few days ago, is the research assisting the drug company in creating the drug that is able to target, or is the drug company working independently in creating the drug that will be able to target?
There can be a happy both ways. So what we do in our lab is we do the research, and then we talk to the, I find that receptor X is highly expressed in this, and then I can go to the drug companies and say, this receptor is really involved in this cancer. Do you have some drugs that might target this that we could test out? And then they can send it to us and we can work together. I'm sure that they're doing research on their end too, but I'm not.
And the only reason, because it seems like there's a lot of unknown that is being discovered. And as it's being discovered in relationship just to the cell, and in this case RNA. So how much of synthetic created chemicals are created, because they're new creations that are not natural to the system, are being to target this based off of the information they got, compared to getting the information and then saying, okay, I can create a synthetic.
Yeah, if we, the first step is to find out what's highly expressed in here. If they have it, then that's spectacular. If we can tell them that that receptor is there, and if you could target this, that would be amazing. Then I'm sure that they'd be very interested in trying to synthesize some drugs that would specifically target that receptor.
And the follow-up, because in relationship to personalized medicine, it gets into, though we're same but different, I guess, as far as at the genetic level, and now at the RNA level, as far as this discussion, that whatever is created is for, again, just a group. It may not be for everyone.
It certainly could just be, and that makes it tougher. It's easier if you say that everybody who has colon cancer can get this drug and they're good. But I think enough people have been touched by it and have had somebody that, the traditional treatment didn't help, that hopefully people would be interested. Hopefully the companies would be interested, or some sort of funding agency would be interested in helping develop drugs that will only help a subpopulation. But I agree, it wouldn't help everybody. It is personalized, but it's not just a person. It's hopefully it would help a subgroup if we could figure out what that subgroup is.
I'll stop with this. But
no, you're fine.
As this is being developed, how much of a new chemical process is being introduced, even in the question of healing in medicine, as the information is being discovered, that changes whatever was originally normal, and that whatever you would use to heal or couldn't heal, now being created specifically to, because they're talking about we're living longer, right? We're living longer because of the drugs we're taking. It's not because we're living longer. That's what I'm thinking. But as we get more information, we'll be living longer because of the drugs we're creating, not necessarily because we're living longer. And the drugs we're creating now, we're adapting to that at that level, at the cellular level. Is that right?
Are you saying that we're using outside drugs to manipulate the cells, and you're concerned about what that might be?
Well, I think it's beyond concern. I'm just trying to understand that this is a process that we're going through.
And it can be, synthetic. Yes, it could be, certainly.
Since it's not
it's not, yeah, it could be not a normal, well, so these cells aren't necessarily normally functioning, you know, in response to your normal chemicals in your body, so you might need to have outside chemicals, yeah. And yeah, so identify new drug targets, and then I'll skip the last thing because I'm going to talk about it on the next slide. So in this experiment, this is a mouse model of breast cancer. And so this is I think it's a biopsy. I don't know that it's a biopsy. It is. This is tissue, breast tissue, and the brown ones are the cancerous cells. And this is with treatment A, and you can see that it cuts down a lot on the cancer cells, but there's plenty left. This is the same original culture, and with treatment B, and you can see that it wipes out a ton more. However, I'm not satisfied. There are still these little cells, and anytime you leave little cancerous cells, they can multiply and they make me uncomfortable. So we could, you know, in this example research plan, we could harvest these cells. We could harvest some of these ones that responded well, and we can find the difference. And why did these ones not respond to that drug? And what else is on these that we could target to either cause them not to divide or cause them to die, kill themselves, basically, lead to apoptosis. So that's an example of, in cancer, the drug resistance that a lot of people face. You could do this treatment and then put on another treatment where it wipes all of them out if we could figure out how to just target these little guys.
Are you saying in this bottom, when the cells that were treated, then sort of, because I think of cancer cells as, okay, they're gone awry, something abnormal, so that these cells are now, based on the treatment, back to a normal cell, or do we assume that
they infiltrated from healthy areas?
Yes.
Yeah, I don't know. I'm not sure in this example, but either way, I guess.
You can still see from those non-cancer cells
Right.
what you're talking about.
Yep. So you would hope that these, and you know what? These ones aren't brown, but maybe they're not back to all the way blue.
Yeah.
You know,
Okay.
so maybe they're still in the danger zone, and they might still need further treatment to really bring it all the way back to a healthy state, or cause them to die. You know, one or the other. So the way that you would do that is you would get these cells cultured, and you could, since they're nice mouse cultured cells, you can, you know, try your different treatments. Or if you want to bring it to, if you can get involved in clinical work, we can get biopsies from patients and keep them alive. And we could actually, you know, we'll do the hard work in the mice, get our promising things, and try, you know, a select few in some of these biopsies and see how they affect the cells in those.
And how long is that really, like, what's the time frame of something like that, honestly, if we get to?
I mean, it depends on the collaborations that you have. I mean, we have access to collaborations with neurosurgeons, and you know, we had to write up why we were doing what we're doing and what we're doing exactly, and it's very labor intensive for patient stuff, which it should be. But I mean, we could try out stuff, like we're allowed to go straight to patient biopsies and try out things like that.
So that could make a difference for somebody like that's awaiting something, there's nothing out there, awaiting something, and within, if they have weeks left, or if
We aren't that yet.
Okay.
We are not. I think that would have to happen more in the hospital.
Okay.
That would be that. Like, we are doing research on. Yeah, it's not really feeding directly back to that patient, but it's feeding back to the general population of, you know, people
Because
suffering
in
from.
the news with the big center that's going over, I mean, all that with the leukemia studies.
Oh,
That's
that's
basically what
amazing.
happened, is it
Yeah.
not? I mean, it really is.
Yeah.
It was his last ditch effort, and they tried something that they thought would happen, and
It was amazing.
It totally was amazing. Yeah.
It is. It's very true. I don't know how his lab, I don't know the connections that they have, and I don't know the difference between, maybe he is an MD.
Right, yeah, no, I understand.
He might be an
what you're
MD,
saying.
and so he might be able to do that. We're researchers, so we can't feed directly back into that patient, but we can feed back into the general,
Yeah. It sounds kind of cool,
information.
though, that we may get to a point where you take a piece of whatever, and we're like, it's gonna take us 3 months, but we'll figure out something to treat your tumor, and you'll be okay, because we can check, like in 3 months, you'll be fine, things aren't gonna change that month, and we can have a directed
Yeah,
medicine for you to get rid of your tumor.
Exactly.
Yeah.
And you might,
Something like that, that's.
I think it's the future of it. I mean, and for me, taking a biopsy of my lung, that sounds pretty bad. But if I have cancer and I got a treatment, and taking another biopsy, at the end of my treatment would mean that we could really make sure that we got it, or, follow up, compared to chemo,
Oh, absolutely.
the alternatives are not the best. So, so that is,
Can I
Yeah.
flip again?
yeah.
This slide brings up about people
Uh-huh.
and what you raised there as far as the cell that didn't get wiped out, even though they treated
Yeah, these guys.
And I'm thinking of what I've seen with people who actually lived through.
Mhm.
So what cell that they had that didn't actually get affected by the virus, not knowing that this process here, the process will have to occur. It's just with the cell as actually an agent for assisting a person to live through it compared to a cell that's dying and trying to, right? It would be the same process.
Yeah, so you could, you take a sample of the cells from the person that survived, and you compare that to a person who is affected by it, and you can see that these genes are upregulated. There's technologies where you can actually sort of turn these cells into these cells. You can introduce a gene expression profile into a cell and give it that resistance, whatever it is, that combination of genes that makes that cell resistant to infection. So yeah, it's absolutely applicable to that.
I was wondering, would this be applicable instead of traditional antibiotics, just infection control? How is that applicable? That's an interesting way of thinking of it.
So this is just another experiment that we ran. It's not cancer. It's not only about cancer. This was I guess you've probably seen this little cartoon on Zoloft commercials or, Prozac commercials. So this is the way that a normal cell works. Nerve A brings the neurotransmitter back into the cell as a way of clearing it so it doesn't overstimulate a normal cell too much. But in depression and anxiety and some other things, you don't have enough stimulation of this, so you can block that reuptake so that you can increase the amount that's hitting the second cell. So that's SSRI, so that's what all these guys are. And so those are good, but they have side effects because they don't just affect here, they have GI effects and a whole bunch of other problems. So it would be really good if we could find stuff that better targets that dorsal raphe, which is this thing again. So this region is more involved in depression, and this region is more involved in panic and anxiety. So, and this is that heat map that I showed you before. It was the LW and the VM, and you can see that they're very different, and there are targets that are different even between those two regions, and hopefully we could find ones that would have less side effects on GI and all the other reasons that people stop taking their SSRIs.
So what we did was we took individual cells from the LW and from the VM. We compared them and we focused on the LW, and we found 3 new receptors that weren't really appreciated before. And we then took this live slice, this is mouse tissue, live slice, and we took the targets that we found directly from our gene expression profiling, and we put drugs that target those receptors right onto the slice, and we were able to show that these cells change their activity. So what happens in a normal person, you have stress, it leads to the LW, and it stimulates these cells up here. In somebody who experiences panic and anxiety, the stress comes in here and you don't have enough signaling out of here. So the goal was to find drugs that would increase the activity of these cells. So in a matter of not too long, we were able to find 3 new drugs that increase the activity of these receptors. And these happen to just be, kind of going to what you were saying, these were ones that existed already from previous research and other stuff. But, you know, I have a whole laundry list of other receptors that are also there that, you know, if somebody could synthesize drugs to it, they might be great too.
So I see that one says oxytocin.
It is, yep.
Okay, so what drug, and I know that's a natural.
I used
name.
oxytocin.
Okay. So what are the drugs that?
So these are the drugs. The receptors are the oxytocin receptor, the lysophosphatidic acid receptor, 1, and this was to the histamine 3 receptor. And actually, this is something that blocks the histamine receptor. So that means normally the receptor is basically being activated by histamine, and it's actually inhibiting it. So if you block that activation, you actually increase the activity. So, and that just was not known before. So, and this actually, I mean, that difference between, these are the action potentials again.
This was, any of this done, and this is more kind of just, this is not human.
This is mouse.
This is still mouse. Okay.
Yep, this is mouse.
But you think this would translate?
I think that it's certainly worth looking at.
Yeah.
Yeah, and this kind of thing could, you know, if we could get a biopsy, or if we could get postmortem tissue from somebody who was suffering from.
Well, there's definitely, I was going to say, I have a very close friend whose mother just had ECT.
Mm-hmm.
And I didn't know that they do there's a special gene that they look for, because all they know is it's related to like regulation, very difficult to regulate meds for depression and anxiety.
Mm-hmm.
And so if she had that gene, it would just mean that they would have to have more targeted
Mm-hmm.
drug therapy for
Mm-hmm.
her. But so that's why it's very, very interesting.
You know, and these are subregions within the same brain region, you know? So it's just, when people are lumping, you know, previously people were just lumping everything together, and you need to break it out into the subregions and then break it into the individual cells, because you find the great stuff when you look at the individual cells. I can't remember, one of these is actually not very high abundance. So if you would have just squished everything together, you totally would have missed it. But, I mean, all of these responses are nothing to sneeze at. They're really affecting the activity of these cells, which would then, you know, increase this output from this region, hopefully back to a normal amount.
This probably has nothing to do with it. I'm always setting up these here trees because I'm sitting here at ignorance.
Okay.
I'm thinking of the energy transfer as a part of the cell expression. This is basically dealing with the biology part of the cell. But it does require a certain amount of energy to make the cell.
I mean, so the brain neurons communicate through electrical pulses, basically. The cell fires, and it sends that pulse down a dendrite or down an axon to another cell, and that's how the brain all works.
And because of the mitochondria, the energy is generated within that in order to activate the cell too. So
yeah.
the effect of I'm trying to think of that electrical process in relationship to the chemical and the biological process as a part of the effect or non-effect to the genetic.
So in this one, so in that electrical pulse, it is run by ions and gradients of them. So sodium, positively charged, is affecting that electrical thing. But the mitochondria, so much happens in a neuron. I mean, they're just changing and communicating so rapidly. And for that protein production, that's where those mitochondria really are important. And actually, where's my little neurons? This cell is coming over and touching this cell, right? And so you can actually, so it's a synapse, that's called a synapse, and at the synapse there's actually a lot of mitochondria, because you don't want to, so traditionally people are thinking, all right, you make RNA here and you make the protein here and then it comes on out, Okay, you know what, this cell needs a response. This cell needs to respond immediately. That stuff happens out here, and that mitochondria is right there to pump that energy out to make that protein happen, to adjust rapidly, or else we wouldn't be sitting here chitty-chatting. It's just, yeah, no time for it to just meander. It needs to happen at the site of action.
And that just brings up in relationship to this whole thing of creating the health process as far as rejuvenating or repairing, how that has an effect, because that message is important, and how much, as we break down the cell, to especially RNA, we really know how much that energy transfer assists in even saying a cell should live or die, or what this should mutate or not mutate. Can that happen? I guess that's what's the process in the brain to have effect on the.
Can the amount of energy available affect the health of the cell?
Right.
Sure, absolutely. I mean, so we study cardiomyocytes, and we study pediatric patients who have major heart problems, and we're finding that there's some serious problems with their mitochondria. So they're not producing energy right. You have a muscle that needs an incredible amount of energy available, and if that energy isn't produced properly, and controlled in the proper way, then you're going to have some problems. So mitochondrial disease is definitely a major problem. Oh, it's 5:30, huh? Yeah, okay. Ta-da. Yeah, so, transcriptomics, one of the huge benefits is that it's high throughput because of the wonderful assays that have been developed. And you can look at the entire expression profile, and using the help of our wonderful bioinformatics people, we can really scan across all of them and look for trends and groupings and pathways that are altered in different diseases or different developmental stages or everything. Single cell transcriptomics, this is my big thing. You can miss a lot of interesting things that when people were just grinding up pieces of tissue, they found great stuff too, but they missed some stuff, and some of it's very important. And then clinically, transcriptomics can be used to find better targets, to test the success of a treatment by taking samples at multiple times during the treatment, and then also find new targets on those drug-resistant cells if, unfortunately, they remain. So I just need to acknowledge all of my nice people. Thanks.
Yeah. It's like a cell is its whole universe in itself. I mean, really, it's just amazing.